So in this section, we are going to discuss about further applications of read mappings. So in the genome, there can be multiple complicated structure variations, like deletions, which is deletions of a segment in the genome, or insertions, that the genome at some particular positions, a new sequence got inserted, or mobile sequence insertions, like retrotransposon, ALU, and tandem duplications or interspersed duplication, inversions, which is inverse of a fragment in the genome, or translocation, which move one fragment from one place to another place. The question is, can you use the approach of mapping to identify these variations? Well, you remember about read pair sequencing that we discussed in assembly. We have read 1, read 2 separated by a distant insert size. It turns out that read pairs is very useful in detecting these structural variations. For example, inversions, translocations, duplication, or transposition can be detected using the mapping approach. We map the first read to the reference. We map the second read to the reference. If we see any changes in directions or the changes in the distance between the first read and the second read comparing to the expected insert size, that is a signal for detecting the structural variations. But if we escape from the genome level, let's move to the RNA level. So it turned out that read mapping can also be used in discovering the splice junctions using RNA-seq. So here, intuitively, you can see that you have a read. Instead of mapping the read to one particular location of the genome, the read can be decomposed into multiple parts, and each part mapped to different regions of the genome. And of course, we have other applications, but it is a good time that you can go out and talk to biologists and to understand what applications can you solve using the BWT or read mappings approach. Thank you.